Proteins are the workhorses of life. They are involved in nearly every biological process imaginable. From catalyzing metabolic reactions to transporting molecules across cell membranes, proteins are essential. Imagine them as microscopic machines, each with a unique shape and function, tirelessly working to keep us alive and thriving. Proteins are made up of long chains of amino acids. These chains fold into intricate, three-dimensional structures. This unique structure dictates the protein's specific function. To study these remarkable molecules, we need a way to isolate them from their complex biological environments. This is where protein extraction comes in. Protein extraction is the crucial first step in understanding how these tiny machines work. By isolating proteins, we can study their properties, functions, and interactions with other molecules. This knowledge is fundamental to advancing our understanding of life itself. Protein extraction is like carefully disassembling a watch to understand how each tiny gear and spring contributes to its function. Just as a watchmaker needs to access the individual components, scientists need to extract proteins to study their individual roles. This allows us to investigate their structure, function, and interactions with other molecules. Why is this important? Imagine being able to develop new drugs that target specific proteins involved in diseases like cancer or Alzheimer's. Or envision engineering crops that are more resistant to pests or drought by modifying their protein composition. Protein extraction is the gateway to these and countless other groundbreaking applications in medicine, agriculture, and biotechnology. However, extracting proteins is not as simple as it sounds. Cells, whether from bacteria, plants, or humans, have evolved sophisticated mechanisms to protect their precious protein cargo. Cells are like fortified castles, protecting their internal machinery, including proteins, within layers of membranes and walls. Eukaryotic cells, which make up animals, plants, and fungi, possess a true nucleus and other membrane-bound organelles. Prokaryotic cells, such as bacteria, lack these internal compartments but still possess a protective cell wall. To access the proteins within, we need to breach these defenses. This is where various protein extraction methods come into play. These methods employ a combination of chemical, enzymatic, and mechanical forces to gently disrupt cells and release their protein content without damaging the proteins themselves. Think of it like carefully cracking a nut to extract the kernel inside. Too much force will crush the kernel, rendering it useless. Similarly, protein extraction methods aim to break open cells gently to avoid damaging the precious proteins within. Section 4. Gentle Persuasion, Non-Mechanical Protein Extraction Methods. Protein extraction can use chemical and enzymatic methods. These techniques dissolve cell membranes or weaken cell walls, allowing proteins to leak out. Solubilization uses detergents to break down cell membranes. Detergents disrupt the lipid bilayer, releasing the cell's contents. Enzymatic methods employ enzymes to break down cell walls. Lysozyme targets bacterial cell walls, digesting them and releasing contents. These methods are gentler, suitable for delicate proteins sensitive to heat or shear forces. Section 5. Forceful Measures, Mechanical Methods for Protein Release. Sometimes, more force is needed to liberate proteins from their cellular confines. This is where mechanical methods come in. These techniques use physical force to disrupt cells and release their contents. Homogenization involves grinding or blending cells to break them apart. Specialized homogenizers create shear forces that rupture cell walls. Sonication uses high-frequency sound waves to create cavitation bubbles. These bubbles implode, generating shock waves that disrupt cell structures. Section 6. From Lab to Life. Applications of protein extraction. These various protein extraction methods provide scientists with the tools they need to isolate and study these essential molecules. Protein extraction has far-reaching implications for our lives. In medicine, purified proteins are used to develop vaccines, diagnostic tests, and therapies for diseases like diabetes and cancer. In agriculture, protein engineering creates crops that are more nutritious and resistant to pests. In the food industry, proteins are modified to improve texture and shelf life. By isolating proteins, we understand their roles in life's processes and open up possibilities for improving human health and agriculture.